is Simon Melov. I'm an investigator at the Buck Institute for Research on Aging. I run the single cell core here. We're deeply involved in multiple projects within SenNet, many collaborations actually across SenNet, but the primary focus of two of the programs here is developing a device to model senescence in tissues, as well as an independent program, a tissue mapping center program, where we try to identify, map, and characterize the frequency of senescent cells in human skeletal muscle and human ovary. It's a big program spread across many labs within the Institute, as well as Northwestern University, which I co-lead the program on the device with Francesca Duncan.
Please give a brief overview of the research you and your team perform.
The mapping program, we work very closely with them in trying to essentially characterize the frequency of these senescent cells in two human tissues, human ovary and human skeletal muscle. The frequency of these cells is currently very poorly understood, actually at a global level in many tissues within the body. And it's got a whole set of major challenges to try and understand how often senescent cells arise in aging tissues, specifically from our perspective, skeletal muscle and the ovary. It's been extremely challenging trying to understand how these low frequency cells develop, what drives them, what the consequences of developing a certain level of them are in the tissue as we get older. But we're making really good progress. And the program overall is very exciting, and I think it's a new phase of biology to sort of map at multiple different levels of the single cell sort of frequency of different types of cells within the body as we get older. And senescent cells are one such type. Two tissues we're focusing on, skeletal muscle and ovary, they've got individual challenges to enumerate and characterize senescent cells as we get older with age. It's generally understood that with aging, we accumulate a low level of senescent cells. And this low level of senescent cells within the tissue has poor outcomes for the surrounding cells. So even though you may only have one cell in 100, which is senescent, it can have a large impact on those other 99 cells, because these senescent cells tend to secrete factors which alter the microenvironment around that senescent cell in a deleterious way. It can actually decrease function and provoke sort of poor responses of the tissue in terms of its overall demand. Now, with regards to the skeletal muscle, we know that there are senescent cells arise with age in human skeletal muscle, and they may impact the function of that muscle. They may cause the muscle to become weaker. They may cause the muscle to perform less optimally as we get older. And in fact, they may even promote atrophy within the skeletal muscle. With regards to ovary, which is the other tissue which we're focused on, the consequences of senescent cells are far less well understood. But it is known that ovary is the first tissue which declines in function in quite a dramatic way with regards to the decrease in fertility within women as they age. Typically, I think most people know that once women enter into their 30s, their ability to get pregnant declines quite precipitously. And that is sometimes referred to as sort of the canary in the coal mine with regards to the heralding the changes which are associated with aging in a bad way. I mean, we know that all our tissues decline in function as we get older, but ovary is one of the very first which declines in function, and it happens at the age of sort of mid-30s or so. And so we're very interested to see if senescent cells play a role in that decline in function. We've already mapped quite a pronounced sort of collection of senescent cells within aging human tissues, ovaries specifically. And now we're trying to characterize where and when those senescent cells arise in the biopsies which we've obtained. We're in a new era. These technologies are enabling us to ask questions in a way where we can actually get answers. The questions have been around for millennia. Why do we get old? Can we do anything about getting old and reduce the deleterious outcomes of the functional decline associated with aging. These are big mysteries which have been around for a very long time, but we haven't had the tools to answer them. We do now, I think. I think we really do have a brand new toolkit with which to answer these age-old questions.
Please share any interesting discoveries you are finding.
What we have found is that it is very heterogeneous. There seems to be a marked degree of heterogeneity within the tissue with regards to specific senescent cells. We're very interested in trying to see if there are subtypes of senescent cells. I don't think it's going to be true that a senescent cell is just senescent and there's just one type of senescent cell. There's quite likely to be many types of senescent cells, and each tissue is likely to have an array of subtypes of senescent cells. And this is one of the technical challenges which we're faced with in trying to implement the overall goals of the SenNet program, is applying these amazing new technologies we have to identify this vast array of subtypes which appear to be common in aging tissues.
What are your thoughts on SenNet as a community and resource?
The SenNet Consortium is a wonderful collection of different expertises, different backgrounds, different technological approaches to tackle these age-old questions. And, it's a really unique collection of institutions and individuals. And every time we get together, I learn something new. That may not be that unusual in the context of these large programs, that people find new things out by interacting with a lot of different people. But in the context of aging, this, I think, is somewhat unprecedented, that this level of focus and multidisciplinary attacks on this age-old problem using state-of-the-art technology is very unique, and it's a really interesting and powerful approach. And I'm really looking forward to what's going to come over the next year or 18 months as we begin to sort of roll all these discoveries together into a coherent look at what's going on in multiple different aging tissues.
What would you tell students and junior investigators who want to research senescence?
People who are interested in getting into the field need to become deeply versed in multiple different aspects of biology as well as technology. It helps to have a background in informatics. It helps to have a background in statistics. It helps to have a background in physiology. It helps to have a background in many, many different areas. And this is why geroscience is a multidisciplinary field. So if you're interested in getting into this sort of brand new area of biology, try and branch out and don't focus on one thing. Do many, many different strands of biology and informatics and statistics. And then you can build tools which will take advantage of some of these amazing new technologies to help further our understanding of the biology of aging and what role senescent cells play in driving deleterious changes associated with age.